there and welcome. I'm Dr. Christopher Pena, the Scientific Liaison for the University of Chicago Comprehensive Cancer Center.
I'm Dr. Mekinda. I'm the Assistant Director for Cancer Education, also at the University of Chicago Comprehensive Cancer Center. And we are back again with some students from our Chicago Eyes on Cancer program, which is a cancer research training program for high school students and college students who are interested in careers in biomedicine. And we also work with high school teachers who are interested in taking a comprehensive research experience, an intensive research experience, back to their students in their home schools.
So we're really excited today. This episode is going to focus on advances in immunotherapy. And we're incredibly excited to have our students because these are basically the future of cancer research, and they're excited to come on today.
Yeah, we know that there are many, many people in the CanTV audience who have been affected by cancer-related diseases, and that's why we are so excited to keep coming back to the studio so we can share more and more about some of the really exciting work that's being done at the University of Chicago to help improve the ways that we prevent cancer and diagnose cancer and treat cancer.
Great.
Yeah.
Well,
So I see your guests are ready to go.
They're ready to go. So let's go ahead and get started. Again, we are Chicago Eyes on Cancer. It stands for Educators and Youth Enjoy Science. We are a 2-year program for high school and undergraduate students that want to participate in biomedical research in the cancer field. You can find us on our website at cancer.uchicago.edu/eyes or email us at eyes@bsd.uchicago.edu or give us a call at 773-702-4678. I am here with my first guest. This is Jenna. Hi Jenna, how you doing?
Good, how are you?
I'm doing all right. Thank you. So today, we're focused on immunotherapy. Why don't you tell us a little bit about what immunotherapy is?
So, in a general sense, immunotherapy is a brand new method of treating cancer. So it works by either activating or deactivating some immune system functions. So, for example, this can be by training your immune cells to attack cancer cells. This can be by blocking the production of a specific protein, or maybe even blocking the activity of immune modulators. And what's really, really amazing about immunotherapy is that it's been very, very effective at treating some of the even hardest to treat cancers, such as melanoma, which is skin cancer.
Awesome. So you had mentioned, it's very good at treating very distinct types of cancers. What are some other advantages of immunotherapy?
So one of the major benefits of immunotherapy is that there are fewer side effects than there are for other treatments like chemotherapy and radiation, for example. And then what's also, in addition to that, is that you can combine it with other treatment methods, so chemotherapy, radiation, surgery, to create a more multifaceted approach to treating cancer. So when you attack the cancer through different strategies, it's more effective at treating the cancer because some cancer cells respond to certain therapies and some do not. So immunotherapy is just another factor you can add on to that to increase the efficacy of the treatment. And another huge benefit is that it's very long-lasting. So this is called Immuno Memory, where immune cells can actually remember how to attack cancer cells, so that if the cancer does resurface, the T cells or the immune cells will know how to attack it, and that way it'll prevent the cancer from further proliferating.
Wow, so we can actually train our own immune system to remember cancer that may come in the future.
Yeah.
Wow. So that sounds great. It sounds like there's a lot of advantages to immunotherapy, but I'm sure that there have to be some disadvantages.
Yeah. Unfortunately, immunotherapy does take more time than traditional cancer treatment methods. So, for example, with surgery, it's a little bit more straightforward. You excise a tumor, whereas for immunotherapy, you have to train the immune system how to attack the cancer. So, as a result, that may take more time. And that also means that you'll have to deal with the side effects of immunotherapy for a more extended period of time. So while they are less severe than other treatment methods, you'll have to work through them for a longer duration. And in addition, immunotherapy can be damaging if the drugs cause your immune system to attack your healthy body organs.
Ooh, okay, so those definitely are some sort of disadvantages. What other challenges are there in immunotherapy?
So only a fraction of patients respond to immunotherapy, and so researchers are trying to figure out why exactly that happens. So for example, in the lab that I'm working at this summer, we're trying to identify mutations of specific genes that cause some people's tumors to shrink while others do not. So what happens is that in some cancers, the cancer cells can essentially masquerade as healthy cells, and that can be very difficult because then if a T cell, which is an immune cell, comes across that cancer cell, the cancer cell will basically communicate to the T cell that I'm a healthy cell. And the T cell will understand that it should not attack that cancer cell when in fact it is cancerous. So what happens is that we've created a drug that's meant to inhibit that pathway, meaning that those cancer cells won't be able to put on the mask of being a healthy cell. So by seeing which patients respond to that type of therapy and which do not, we can find patterns within their genes, so that in the future with future patients we can see whether they have that gene or not, and that way we can further specialize the treatment for their specific genes. This is called personalized medicine, and it helps create a more targeted approach because, as you know, every patient is different.
Awesome. Well, I think we're going to get a little bit more into personalized medicine later on in the show, and we'll have you back on.
Yeah.
But for now, thanks for talking about your work and for describing us a little bit about what immunotherapy is.
Thanks
Thank
for having me.
Awesome. So again, we are Chicago Eyes on Cancer. We are a research program for high school and undergraduate students that want to get experience doing biomedical research in the cancer field. Again, a 2-year program. You can reach us at cancer.uchicago.edu/eyes, or give us an email at eyes@bsd.uchicago.edu, or give us a ring at 773-702-4678. I am here with Blake.
Hey,
How you doing, Blake?
good.
To see you. Awesome.
Good to
So,
see you as
the subject has been immunotherapy. Just kind of scaling things back a little bit. Can you tell us about the kind of immune cells that are out there?
Yeah, I can do that. There are 2 main kinds of immune cells. There is neutrophils, and then there is macrophages. Neutrophils are the first line of defense your immune system has. They fight all the harmful bacteria and fungus. Yeah.
I think fungal infections.
Fungal infections. Yeah. And then there's the macrophages. Those are the ones that like to store up and store into the memory bank, the immune system memory bank. And as you mentioned earlier, they like to remember what diseases attacked and then store it and prevent them from attacking again.
Okay, so I think that's why
Yeah.
some people, when they get sick, the first time takes a long time to recover,
Exactly.
but then the second time they can either completely bypass getting ill, or in some cases just get ill for a couple days.
Exactly.
But bam, it's all
Exactly.
taken care of. So in terms of cancer and macrophages, what sort of relationship is there? What kind of macrophages deal with cancer?
So there's 2 main kinds. There's cancer promoting and then there's cancer killing. So there's M1s. These are the cancer killing macrophages. These ones go in and try and dismantle the cancer as best they can. Then there's M2s that are mostly used for repairing cell damage and more housekeeping kind of duties. And M2s promote cancer. So because they're not killing it, they're helping it. They're using it as a disguise to proliferate even more and spread and become more serious.
Wow. And so the research that you're doing in the lab as part of this program, what does it have to do with macrophages?
So in our lab, we work on the effects of obesity on these macrophages. And as we established earlier, the M1s and the M2s is a paradigm, and they usually, and most macrophages fall within this paradigm. But our lab's working with metabolically activated macrophages that are activated through palmitic acid, or the fatty acid in your body. We've tried to like analyze and put together this pathway with palmitic acid and how it affects these macrophages. And we've seen, our data shows that palmitic acid actually induces the metabolic phenotype, which is not good because unlike the M1s, they're very similar on the outside, but in the inside they're very different. And M1s can kill cancer because they're armed and
Ready to go.
But yeah, exactly. But then metabolically activated macrophages, they're more used for moving and storing fat. So if you put a bunch of soldiers, lack of a better word, that are used for moving, you really can't attack the cancer. And that we're finding that tumors grow when patient is obese.
Wow. So kind of going back to that, it seems that this palmitic acid, this high-fat diet, almost in a way cripples that army you were talking about. So you don't have the same M1 macrophages fighting cancer. You're sort of putting them, you're training them to do something else.
Exactly.
Awesome. So I think that's it for your research.
Yeah.
Yeah.
Future. Let me just
Yes, go ahead.
in the field. In translation from bench work to like clinical, this could, if we can actually elucidate this pathway, we can pinpoint the NOx2 system, which is what we're trying to, which is a complex that produces reactive oxygen species that we think is responsible for changing this phenotype to metabolically activated. If we can target that specifically, we can turn these metabolically, these useless immune cells into useful immune cells and attack cancer directly in diseases such as triple-negative breast cancer and type 2 diabetes.
Awesome. That sounds good. So it sounds like if this research works out, this will really be good for people that are...
Be good for a lot of people.
This is awesome. Well, thanks for stopping by.
No problem.
We will see you in a little bit. Again, we are Chicago Eyes on Cancer. We are a 2-year program for students that want to do biomedical research in the cancer field. You can reach us at cancer.uchicago.edu/eyes. Email us at eyes@bsd.uchicago.edu or give us a holler at 773-702-4678. I'm here with my next guest, Ocean.
Hello.
Ocean, welcome back. It's good to
Thank
see
you.
you
It's
again.
been a while.
I know. So, we've been talking a little bit about immunology research. Can you tell us about what you're working on in the lab?
Yes, so I work with T cells, and basically what T cells are they are these soldiers of the immune system in which they either indirectly or directly will kill foreign invaders or unhealthy cells. So I work with these with a mouthful called V gamma 9, V delta 2 T cells, and basically we're trying to figure out how these T cells are activated. So these T cells are different than traditional T cells in which they require a phosphoantigen to activate instead of a MHC molecule. And basically what we know so far is that there's this requirement of this protein called BTN3A and this phosphoantigen in order to activate this T cell. But we don't really understand what's going on, what is the molecular mechanism behind this activation. And if we're able to figure out the activation molecular mechanism, then we can figure out how to improve immunotherapy techniques such as CAR T cell therapy.
Awesome. So, you had mentioned working with different types of T cells, namely the alpha beta as one, and then gamma delta T cells. So, given these really long names, there has to be some
Welcome
kind of
to
difference.
immunology.
Wow. So there has to be some kind of difference between these types of T cells. What are they?
So the first difference is that alpha beta T cells have a T cell receptor composed of an alpha and beta chain, whereas gamma delta have one composed of the gamma and delta chain. The biggest difference to really think about is this requirement of MHC molecule. This is extremely important because in cancer, MHC molecules tend to be downregulated. This is for most cancers usually too. And while alpha beta T cells are the most common type of T cell in our body, if MHC molecules are downregulated, how are we supposed to activate this T cell? Like, how is it supposed to be doing its job? Whereas gamma delta T cells don't require this MHC molecule. Instead, they require, in my case, a phosphoantigen or a protein. And this is important because, again, to this activation issue is that also, like, cancers can actually make a receptor or whatever type of antigen in order to turn off your T cell. So we haven't really been seeing this with gamma delta T cells since gamma delta T cells have actually been inside the tumor.
Wow. So, a little bit more specifically, you had mentioned V gamma 9, V delta 2 T cells. So why are you interested in that particular population?
So this is one of the more well-studied gamma delta T cells. It's about 1 to 10% in peripheral blood, so it's one of the more common ones. And as I mentioned, gamma delta T cells are not common as it is, so this is awesome if we can study the most common one in your blood. But really, I want to talk more about this CAR T therapy. So CAR T is basically when you take T cells from your body and they're going to culture your T cells. And basically what they're going to do is they're going to engineer in some way in order to specifically attack your tumor. So as Jenna had mentioned, this is a form of personalized or targeted therapy. One problem with CAR T is that although they have success in leukemia, such as blood cancers, they traditionally use alpha beta T cells. They're very expensive. They're about $600,000 and very time consuming.
Wow.
Yes. So what's nice about our T cells, since it's most common, it doesn't require this MHC molecule. So for example, your MHC molecules are way different than my MHC molecules. So you kind of, we at least think so, will remove this donor compatibility issue, which will which will also reduce the amount of time that it takes to make this therapy, in addition to reducing the costs. And cancer, as we have mentioned before, is very expensive. So we're very interested in this T cell, and we really just need to understand the mechanism behind activating this T cell, because if we don't know how to activate it, then how can we use it for CAR T?
Right. Wow, so it sounds like your research not only is going to help people, but it's going to hopefully save them a lot of money too.
Hopefully.
Awesome. Well, thanks for stopping by again. We'll see you a little bit later. And again, we are Chicago Eyes on Cancer. We are a 2-year program for high school and undergraduate students that want to participate in biomedical research. You can reach us at cancer.uchicago.edu/eyes. Email us at eyes@bsd.uchicago.edu or give us a call at 773-702-4678. Our last guest is Jaime. How are you doing today, Jaime?
Good. How are you?
Awesome. So I think today you're going to talk to us a little bit about castration resistant prostate cancer. Why don't you tell us about what that is a little bit?
Okay, yeah. Prostate cancers are hormone dependent. Specifically, they are androgen dependent, which means that this is the hormone that is driving this disease. And the first line of treatment is to blockade that hormone. But once the cell becomes resistant to this hormone deprivation therapy, then it's classified as castration resistant.
Great. Okay, and so how does your lab tend to incorporate personalized medicine in this fight against this castration-resistant prostate cancer?
Yeah, well, as you know, personalized medicine is the treatment of a disease. In our case, it's cancer, in a case-by-case approach, since, two people aren't alike. No two people are alike, so therefore their treatment and their style of therapy should not be alike either. That is the approach that is taken to this cancer, to their respective cancers, and this kind of medicine tailors therapies to the genetic and the epigenetic risks of a certain person, and that way the therapy that they're being given is more efficient, and it is lacking all of those setbacks that non-specific and all those non-specific side effects that therapies like chemotherapy can have.
Oh, great. Okay, so I hear that you work in a translational science lab. So what's the advantage of making a discovery in that kind of lab as opposed to one where you're strictly just looking at, let's say, like DNA structure all day?
Yeah, well, my principal investigator is Dr. Schmolowitz. So he emphasizes how our work on the lab bench can really translate into the work that he does with his patients. And a breakthrough in our lab would mean a different type of therapy for prostate cancer patients in his everyday work.
Great. So it sort of sounds like, medicine these days is not one size fits all. It's specific for every individual person out there, especially for a disease like cancer.
Definitely.
Great. And I think you're making a valuable contribution in that regard. So thank you very much.
Thank you.
All right. Stick around. Again, we are Chicago Eyes on Cancer. We are a biomedical research program for high school and undergraduate students that want to get a good lab experience and really want to learn a lot over a summer, over 2 summers actually. You can find out more about us at cancer.uchicago.edu/eyes or give us an email at eyes@bsd.uchicago.edu or give us a holler at 773-702-4678. We'd be glad to talk to you. So welcome back, Jenna.
Hi.
And then Jaime, we saw you a second ago. So you both are first-year students in this program. And so I think I'll start with Jenna first. What has this experience been like as a first-year student for you?
So for me, I love the variety of experiences I've been able to get exposure to. So for example, today I've been really busy. This morning I spent like an hour in my lab working on my experiments. Then I spent like 2 or 3 hours today. I got to shadow my principal investigator. He's a doctor at the University of Chicago's Medical Center, and I got to shadow him for a little bit. Then after that, I went to our weekly Wednesday meetings for the program, in which I got to hear about one of the university's esteemed researchers and her work with colorectal cancer. So that was really amazing to get the chance to hear about some groundbreaking research that's going on not too far from where I live. Wow. And then finally, I'm here in the recording studio. I getting the chance to do such an amazing opportunity, especially as a high schooler. It really means a lot to me.
Great, awesome. Jaime, what do you think about being a first year in this program?
It's been an amazing experience being a first-generation Mexican-American, meeting that I don't have the mentors that some other students might have. So the opportunities that have been given to me through this program, like the career talks on Fridays, the lectures on Wednesdays, they've given me this entire network of people that I would have otherwise never even crossed paths with, and now they're just an email away, and I think that's just amazing.
Awesome. Great. So I would say mentoring has been big for
Yeah,
both of you.
definitely.
And of course the variety of experiences that you get. And so
Yes.
we hope to see you guys again next year.
Yeah, definitely.
Our second year students. So thanks for coming by.
Thank you.
And we're going to bring some second year students. But first, we are Chicago Eyes on Cancer. We are a training program for high school and undergraduate students that want to participate in biomedical research in the cancer field. You can reach us at cancer.uchicago.edu/eyes, email us at eyes@bsd.uchicago.edu, or give us a ring at 773-702-4678. So I'm here with some second-year students. Welcome back. Welcome back, Ocean. Welcome back, Blake. Now, one unique thing about Eyes on Cancer is that you can stay within the same lab you work in over the summer, the next year, or you can switch labs. And so, Blake, you've actually stayed in the same lab.
Yes, I
So
have.
why don't you talk about the advantages of that? And then, Ocean, you can chime in afterwards and tell us what it's like to switch labs.
So I work in an immunology lab, and it's been really nice. Everybody in there has been really friendly, genuine, always offering help. It's been a really smooth transition from having no lab experience to now going on 2 years of lab experience. So what I'll say is when you stay in the same lab, you build relationships with your lab mates, and they're a little bit deeper than they would be if you switched, because you have more time with them. Also, you start to know where things are in the lab. You start knowing where people's rhythms are, what projects they're working on. You can get a lot more invested into that specific lab, and I'm happy with it.
Great, awesome. Ocean, what is it like to switch labs?
So the first lab that I worked with gastric cancer, specifically with cell culture. And during that time, I was a high school senior, so I was thinking about what do I want to major in? Where do I want to go to college? I was growing up. It was
Awesome.
weird. So anyways, we had a lecture by my current PI called Dr. Erin Adams, and she combined biochemistry and immunology. And I was very interested in biochemistry, but I didn't know if it was for me. So I went ahead and sent her an email. I was like, hey, can I meet up with you? Can we talk about biochemistry? And she's like, sure. So she put an hour aside for me. She talked to me a little bit about her lab, in addition to what a major in biochemistry is like. And for me, it was like so eye-opening because I thought I was just going as a biology major. But I didn't realize that biochemistry is really understanding the mechanism behind things rather than just what they're doing. So I went ahead and majored in biochemistry in addition to switching her lab. What's great about switching to her lab was that not only did I learn cell culture, but I learned a whole different side of techniques that are directly related to my major. So I was learning things like PCR, Western blots, and protein purification, how to do stuff like that. And what's nice about switching labs, I think, is that you learn a variety of techniques. Whereas you may not have this in-depth relationship with your mentor, it's kind of really good for learning things that maybe more in your field, or maybe you want to switch labs in order to get a feel for a dry lab or a wet lab. Also, what's great about switching labs is that I have 2 PIs, so 2 people that I networked with, and I will be attending the University of Pennsylvania in the fall, and I want to get into a lab because I love working in a lab. And what's great about having these 2 PIs is that they're using their connections and kind of talking to people at those labs and see if I can get into that lab too, and kind of giving me this recommendation.
Awesome.
So, yeah.
Great. So it sounds like depth or breadth, either way, is a good experience in this program. So thank you guys for stopping by today. And I think that's it for us. So again, one last time, we are Chicago Eyes on Cancer. You can find us at our website at cancer.uchicago.edu/eyes. Email us at eyes@bsd.uchicago.edu, or give us a ring at 773-702-4678. We're a program for high school and undergraduate students. Anyone out there who's interested and wants to do cancer biology research in a lab, whether it's a dry lab setting where you're doing more work on the computer, or you want to work at an actual lab bench, handle reagents, materials, all that kind of good stuff. So thanks for stopping by.